Welcome to the webinar. We're doing dimensional reduction and clustering in FlowJo. My name is Josh Luthy, and I'm an application specialist here at FlowJo. If you guys have any questions throughout the webinar, feel free to just type any questions in the chat, and I'll try to answer those as we go along. So here's our agenda for today. We're going to just do an overview of dimensional reduction and clustering with a focus on UMAP and Phenograph. We'll look at some comparisons as well to t-SNE and FlowSum. Then towards the end, we'll jump into FlowJo and we'll take a live demo, an overview of a quick setup of some of the plugins, like where to get those, setting up R. Then I have an analysis we can look at. And then there's a new tool I'd want to show you guys. It's called Sleepwalk. It's an R package. And it's a really cool way to take a look at clustering and embedding results you have and kind of get a better idea of how well does that really represent your data. So we'll take a look at that at the end. So as most of you are familiar with Flow, I'll focus today on the data analysis aspect, particularly dimensionality reduction and clustering, which are key to interpreting high-dimensional flow data. So as you know, flow cytometry generates high-dimensional data, often with dozens of parameters. Dimensionality reduction techniques like t-SNE and UMAP are essential for visualizing and understanding this complex data. And understanding how to analyze this is crucial for researchers and professionals in various biological fields. Again, today we'll focus on key data analysis aspects that underlie effective interpretations. And what we see here across here, some of these examples are some of the methods that are available in FlowJo to perform dimensionality reduction. Keep in mind, these are just some of the methods. There are additional ones that are available for download as a plugin from the FlowJo Exchange. So dimensionality reduction in general, the goal here is to create a low dimensional representation of our high dimensional data set that preserves the overall structure of the data as much as possible, right? The method, this method here, t-SNE that we're showing, and others help us reduce the complexity of the data while preserving the most critical information, enabling us to see patterns and relationships that might otherwise be missed. Dimensionality reductions help give us a clear understanding of the data and can lead to more impactful biological insights, and it's vital for visualizing our high-dimensional data. And here with t-SNE, by reducing dimensions, we can better comprehend intricate correlations within our data. Dimensionality reduction, again, attempts to group events with similar multi-dimensional expression patterns together within the dimensionally reduced data space. This helps to condense complex data while retaining its essential information, making patterns visible. Like we can see in this example here, where we can clearly see the different populations of cell types that are overlaid on this t-SNE plot. Talk about the power of t-SNE, some of its basic principles, advantages, and limitations. So t-SNE, or T-distributed stochastic neighbor embedding, is a popular dimensionality reduction technique. It's widely used in Flow for visualizing our high-dimensional data. It was developed by Lawrence Vanderbieten and Jeffrey Hinton. It's designed to capture local structure of the data by bringing similar data points closer together in the low dimensional space, typically 2D or sometimes 3D representations. And t-SNE works by converting high-dimensional Euclidean distances between data points into conditional probabilities that represent similarities. The algorithm then tries to minimize the divergence between these probabilities in the high-dimensional space and the lower-dimensional map. Basically, it aims to ensure if 2 points are similar in high-dimensional space, they're going to remain close to one another in the reduced dimensions. One of the key strengths of t-SNE is its ability to create those clear, visually interpretable clusters or islands, right? Groupings of events in the low dimension space. And that makes it especially useful to identify distinct cell populations. By reducing the dimensionality of the data, t-SNE again helps us to reveal underlying biological structures such as cell subtypes or activation states in a more intuitive manner. It's really highly effective at capturing local structure, so meaning it excels at revealing small clusters or subtle differences between cell populations. And its ability to produce visually clear and separated clusters does make it a powerful tool for exploratory data analysis and flow. t-SNE is also nonlinear, so this allows it to handle complex nonlinear relationships that often exist in biological data. However, t-SNE is not without its limitations, right? One major drawback is its computational intensity, in particular with large data sets. So this can lead to a long processing time. And another challenge with t-SNE is its tendency to lose the global structure. So while it's great at revealing those local relationships, it can distort the overall data landscape, making it difficult to interpret global patterns or relationships between those islands or clusters that we see. Additionally, t-SNEs, it's also sensitive to parameter choices, such as perplexity, learning rate, right? Those tunable parameters that you see, like if you initiate t-SNE in FlowJo. So fine-tuning of those parameters can be tricky, and it might require multiple iterations. However, in FlowJo, we do offer OpsNE, and OpsNE, it does aim to kind of simplify those choices for you. So when it's calculating, it attempts to optimize some of those parameters during the calculation process. For instance, it might not go the full number of iterations if it finds that it's not changing significantly between each round of calculation. So, and finally, t-SNE does not inherently preserve those distances between clusters. So the spacing between clusters or islands in the output may not really reflect their true relationship in the high dimensional space. So next, let's talk about UMAP. So UMAP, oops, sorry, I wasn't paying attention to the chat. So sorry, someone's asking slides to be available after the webinar. Yes, those are always available from our website. You can download slides and watch the talk again if you want. Someone asked, does splitting of cluster of the same color, example, the pink or blue one means unique subsets? So yeah, if we go back, so yeah, like this, the B cells or the NK cells here, there's some split between those islands. Yeah, I mean, this is just overlaid with some general lineage gates, but we can see, at least based on t-SNE, right, there's some more definition there. So it's capturing more of the local structure of the data and separating those spaces. So if we did additional gating, maybe down into further subsets of those, we would be able to determine different subsets within those. Okay. So back to UMAP, uniform manifold approximation and projection. And so this is more recent dimensional reduction technique developed by Leland McInnes and John Healy. And it's grounded in concepts from topological data analysis and manifold learning. It basically works by constructing a high dimensional graph of the data representing the local relationships between data points, and then it optimizes a low dimensional representation that tries to preserve these relationships as best as it can. The key idea here is behind UMAP is to maintain both the local and global structures of the data, allowing for more accurate and meaningful visualizations. So here we see some of the tunable options that are available when you initiate UMAP plugin in FlowJo. So there's nearest neighbors, minimum distance, and number of components. So with a nearest neighbors of something low, like a number of 2, we see that UMAP merely just kind of glues together some small chains, but due to the narrow view, it fails to really see how those connect together any further. And this represents the fact that from a fine detail point of view, the data is very disconnected and scattered throughout the space. As neighbors start to increase, UMAP manages to see more of the overall structure of the data, and it glues together more of those components to better kind of convey the broader structure of the data. And so by the time we reach, neighbors of around 20, we have a fairly good idea of the overall view of the data, showing how various colors interrelate to each other over the entire or the whole data set. If you go further, right, more focus is placed on the overall structure of the data. For example, like if you go up to something like nearest neighbors of 200, right, we might get a plot where the overall structure is well captured, but at a loss of some of the final local structure. And then minimum distance. This controls how tightly the resulting UMAP plot is allowed to pack points together. So quite literally, it just provides the minimum distance apart that the points are allowed to be in low dimensional space. So this means that low values of a minimum distance will result in tighter groupings of events. This can be useful if you're interested in clustering or in finer topological structure. Large values of that minimum distance will prevent UMAP from packing points real tight on top of one another, and will focus on the preservation of the broad topological structure instead. And then number of components. This you can choose like how many components or parameters do you want to get back from UMAP? Like if you choose 2, you get it like an X and a Y, right? A UMAP 1 and a UMAP 2. Or you could choose 3, you can get an X, Y, Z, then you can plot that in a 3-dimensional type of view. Or you could go even up, 10 different components. So how does this kind of differ from t-SNE? So one of the major differences with UMAP and t-SNE is, again, UMAP's ability to preserve more of the global structure of the data, while t-SNE focuses heavily on local relationships. UMAP strives to maintain both that local and broader global relationships, giving you a more accurate sense of how clusters are related to each other. This means that UMAPs can provide a better overview of the data's overall structure, helping to understand relationships between different clusters or populations. Yeah, someone asked in the chat, is there a typical minimum distance used for UMAP analysis? So yeah, the default is 0.5. I believe that's the default in the plugin. Um, and as we'll see later, UMAP's pretty forgiving and um, performed pretty well with a lot of those default settings. So like that default value for a 0.5 tends to work pretty well. Like all these plots we see here were created with that default minimum distance of 0.5. UMAP is also really highly scalable, so it can handle large data sets more efficiently than t-SNE. So it does make it kind of a preferred choice if you're dealing with extensive data, really big panels, high number of events. So that computational efficiency is really a significant advantage to UMAP, as it typically runs faster and requires less memory compared to t-SNE. Again, especially when you're working with large data sets. And another advantage is, again, the simplicity in terms of those parameters that we talked about, those settings there. Unlike t-SNE, where it requires maybe careful tuning of those parameters like perplexity and learning rate, UMAP typically works really well with just those default settings. When that reduces like the need for a lot of extensive trial and error runs. Sorry, I was checking the chat. So in flow cytometry, right, UMAP is increasingly being used and adopted due to its ability to preserve both local and global structures, which can be crucial for accurately identifying and understanding complex cellular relationships. Right? Researchers might use UMAP to explore cellular heterogeneity, track changes in cell states, or identify rare cell populations that might not be visible with traditional gating strategies. And in FlowJo, it's easy to integrate UMAP into your analysis. And so that's a great way to get this powerful tool for reducing dimensionality and uncovering those insights. So just a quick kind of comparison and overview again. So t-SNE is, again, particularly effective at capturing and preserving the local structures in the data. And so it excels at separating those small, closely related clusters, what makes it ideal for identifying subtle differences between populations. t-SNE is widely used and validated in the field, so it does make it a trusted tool for many researchers, particularly when analyzing complex high-dimensional data. Some of the cons of t-SNE, you do lose some of that global structure. So there's a tendency for t-SNE to distort that somewhat. While it excels at that local clustering, it can lose that overall relationships between those clusters, which can lead to misinterpretation of how these are related to one another. It's also, again, computationally intensive. So especially with large data sets, the processing time can be long. The algorithm requires significant memory. And so that can be a bottleneck if you're doing a large type of analysis. It's sensitive to parameters. So results of t-SNE, again, can be sensitive to how you input or tune those parameters, like learning rate and perplexity. So finding the optimal settings might require some different iterations, or asking some other experts that might have used it in the past. And now looking at UMAP, It's a more balanced approach by preserving both local and global structures of the data. It's highly scalable and efficient, so it runs faster, can handle larger data sets better than t-SNE. So this makes it a practical choice if you're analyzing a large data set. Again, those parameters that you input to the algorithm, it's less sensitive to those parameter choices compared to t-SNE. So it generally performs well with those default settings. And this can reduce the need for excessive parameter tuning, and it makes it just easier to use, especially for those that might be new to using dimensionality reduction. Some of the cons, though, while UMAP is good at preserving the overall structure of the data, it can sometimes produce clusters or groupings that are less distinct or more overlapping compared to t-SNE. And so this can make it harder to visually separate and identify those small, closely related populations. UMAP's somewhat less established. It is a little bit newer technique, but it is gaining traction in the field and rapidly becoming popular. It may not yet be as widely validated or trusted as t-SNE. So in some cases, though, t-SNE might be preferred, right? If your primary goal is to explore and visualize local structures within the data, such as identifying small, really distinct cell populations. UMAP might be preferred if it's important to maintain global structure of the data or understand those broader relationships between those clusters. And that can make it ideal for exploratory analysis and understanding overall patterns in your data. Or if you're working with large data sets, again, UMAP could be benefit there because it scales well and it runs faster. Okay, now we'll talk about some clustering. So once we've reduced dimensions in our data, clustering becomes the next crucial step, right? It's going to allow us to group similar cells together based on their expression profiles, helping to identify cell populations and subpopulations. So clustering methods like FlowSum, Phenograph, each have their strengths and are used to define these groups in a way that is biologically meaningful. So here we're looking at FlowSum. So FlowSum is a powerful clustering algorithm. It's specifically designed for the analysis of flow cytometry and mass cytometry data. So it combines the strengths of self-organizing maps with hierarchical clustering to efficiently identify and organize cell populations in our high-dimensional data sets. And the core idea behind FlowSum is to map high-dimensional data onto a lower-dimensional grid. And this is where similar data points are placed closer together. And then this is achieved through the self-organizing map, right? That grid pattern. And it's a type of artificial neural network that clusters the data based on similarity. Then those maps are essentially reduced to preserve the topological relationships between data points. And as the sum or the self-organizing map learns, it adjusts those nodes so that similar cells are grouped together in the map, forming a pattern that reflects the underlying structure of the data. And after that SOM has been trained, FlowSum performs now the second step using hierarchical clustering. And this step groups those nodes on the map into larger clusters, creating a hierarchical tree that organizes the data into those broader cell populations. So the combination of the sums with the hierarchical clustering allows FlowSum to identify both fine-grained and broad cell populations, making it a versatile tool in FlowJo. And so that's what you'll notice when you start FlowSum, there's kind of two options there. There's like a grid size, that's 10 by 10. So that's the first 100 clusters that it makes onto the grid. Then there's the option for meta clusters, and that's where you'd want to input a number of clusters you might expect to get back from the data. And then it further uses hierarchical clustering to refine that grid into the smaller number of meta clusters. And someone's asking if there's any tips on picking the number of meta clusters to start with FlowSum. That's a tricky question. There's not really like the best method. And that I would say is like one of the downsides of FlowSum, because you have to tell it how many clusters to return back. Some people will run like a different type of clustering algorithm, like Phenograph or XShift, because those will return back however many clusters that they find within the data. And then say if you get back 20 clusters from Phenograph, someone then might take that into FlowSum and ask for 20 meta clusters, and then see how that performs with their data. And then with the additional outputs that you get, right, the self-organizing map and the heat maps, that can help you kind of interpret those results more so than just like Phenograph, where you're getting clusters. However, there are other tools, right? If you wanted to make those kind of heat maps from Phenograph, it's just pretty convenient that it happens directly in FlowSum. That's one method. Or, if you've done some manual gating and you've kind of already identified populations from your manual gates, that could inform you how many meta clusters to ask from FlowSum. And then you kind of compare clustering from FlowSum against your manual gating approach. But FlowSum really has become a go-to method in flow cytometry due to its ability to efficiently handle those large data sets. Again, it's well suited for data where there's large number of markers or cells, or where traditional gating strategies might struggle to capture the full diversity of cell populations. Again, one of the benefits there is its speed and scalability. So the algorithm is really computationally efficient, allowing it to process large data sets much faster than some other clustering methods. The hierarchical clustering step in FlowSum also helps to ensure that even small, distinct populations are captured. So this right, can be critical in studies where you're detecting rare events. So in practical applications, FlowSum can be used to explore immune landscape, identify biomarkers, characterize cellular heterogeneity in various disease states, including cancer and autoimmune diseases. Again, another advantage there is its intuitive visualizations. So the SOM grid provides a clear and interpretable map of the data. So it can help you to understand your complex data set and interpret how the clustering performed. And a pro tip there, when you're in FlowSum in the plugin UI, there's an advanced button there. If you click that, there's some additional options there, like controlling the colors of your outputs. But one is to create a summary plot, and it generates a PDF, and it does like all of the available visual outputs from FlowSum. So different kinds of heat maps, different kinds of self-organizing maps. It actually runs a t-SNE in R and then overlays all the clusters onto that. So it does take a little bit of more time because it is doing that t-SNE calculation in R. But the outputs that you get in that PDF are pretty nice. So if it's the first time kind of exploring your data, you might want to give that a try, generating that summary PDF. Someone's asking, running Phenograph always failed for them. The possible reasons and what's possible solution. Possible reasons. First, I would just check with making sure the R packages are installed. So in the PDF that comes with the download, it lists packages, so you can take that command, put it in R, make sure those are installed. If that's fine, then I would check, where are you saving the data? Make sure there's not any problems with Flojo being able to read, write information, or R for that matter, to read, write data to certain locations, right? If it's on a server or something like that, try locally or try to save as a Flojo archive, run it from the archive, see if that works. That might inform you, if it does work from an archive, that maybe there's some kind of permission issue with where your workspace is saved, if it's on a server, for example. Another possibility is the amount of data you're throwing at it. I mean, it is more computationally intensive than FlowSum. So if you try running on a smaller subset of events, like does it work with the small number but fail on large data, then maybe you're pushing the amount of RAM or compute power you have on your computer. So those are some things to check. But all these plugins will output results in the output folder. So look for R script, Phenograph text file. And if you have that text file, you can just send that in to flojo@bd.com, and then I can take a look at it and help you from there. It's kind of the easiest way, is just to see what's happening from R. So that rscript.phenograph.txt file has that information. So onto Phenograph. So Phenograph is an advanced clustering algorithm, and it's designed specifically for high-dimensional single-cell data. It was developed by Jacob Levine and colleagues, and Phenograph is well suited for identifying distinct cellular populations within complex heterogeneous data sets. And it really excels at finding rare cell types and subtle differences between cell populations that might be missed by traditional clustering methods. The core idea behind Phenograph is it constructs a graph or a network where each node represents a cell, and the edges connect nodes that are similar to each other based on their high dimensional feature profiles. So it begins by building a k nearest neighbors graph. And in this graph, each cell is connected to its k nearest neighbors, and it forms a network that captures the local structure of the data. And then from there, it applies community detection technique, specifically Louvain clustering method. So it applies that to this k nearest neighbors graph. And this Louvain method is an optimization algorithm, and it's used to detect communities or clusters in these networks. So through this process, Phenograph can identify those groups of densely connected cells within the graph, which correspond to those distinct cells, populations, or clusters. So the result is a really, really good and robust data-driven clustering that can uncover those complex relationships between cells. So one of the strengths with Phenograph is its ability to automatically determine the number of clusters without requiring the user to specify this parameter in advance, which is a limitation in like FlowSum, for example. Phenograph has become a pretty popular tool in flow cytometry analysis, again, due to its effectiveness in handling high-dimensional data and its ability to detect those rare cell populations. So in Flojo, it's easy to integrate Flojo into your analysis, right? Installing that plugin, its ability to handle complex data makes it useful for those large scale studies where traditional gating might be insufficient. Once we have our dimensionality reduction, our clustering done, we often go into Cluster Explorer, and this is a tool that allows us to load in those results there. Dimensionality reductions different. You can load in multiple, so you could load in UMAP, t-SNE, tri-maps, different clustering results, and even in the latest release that's in Flojo 1010, you can choose to load in any of your manually gated populations, which is pretty cool. So instead of just having a cluster, if you just have UMAP and you want to look at your gates with Cluster Explorer and do this type of analysis, you can do that just from your manual gated populations. So in summary, real quick, dimensionality reduction and clustering are complementary techniques. So again, we're going to use both those when we're doing a high dimensional analysis. And both t-SNE and UMAP produce biologically meaningful embeddings, with t-SNE generally preserving more local data structure and UMAP preserving more of the global structure. While the dimensionality reduction techniques like t-SNE or UMAP help us to visualize the data in a more intuitive way, clustering helps to make a sense of these visualizations by identifying which cells are similar and belong in a group or population together. So again, in Flow, these techniques are crucial for translating complex multidimensional data into actionable biological insights. There are some challenges still, like oftentimes the next step, people want to annotate those clusters. So having a nice method for biological annotation is a still challenge that there's some options out there, but we haven't implemented anything yet in Flojo. We're looking towards some type of annotation, interactive visualizations, and thorough comparisons with other methods. So areas like that can be improved upon. Okay, so let's take a demo. So starting with just real quick, like R setup, plugin setup, things like that, right? Right now I'm on a Windows machine. My Mac died, so I'm waiting for a replacement, which I'll be happy to get it. This thing's pretty underpowered. So we'll see in Flojo. I have an analysis that's already done because this machine just really won't work well for me. But so when you're installing R for Windows or a Mac's pretty simple. Windows, recommend going with the default installation. It typically will go into program files. Sometimes it might go into on your local machine app data. So like under your user's local account, there's an app data folder. That's fine too. You'll want to install R tools for whatever version of R you have. So the latest one is 4.4. And again, just running through with the default installation for that is the best way to get through that. R is going to need R tools to compile some packages and make sure you can install libraries that it needs. And then any of these plugins you can get from Flojo Exchange. So UMAP, FlowSum, Cluster Explorer is now native to FlowJo. If you're in the latest version 10.10, you won't even really need this plugin, but it's available for like older versions. But when you download these plugins, they'll come with a zip file. You can extract from the zip, and it'll have a PDF and the jar file. You're gonna take that jar file and put it into a folder. So like this is where my plugin folder is located on this machine. It's on my user account OneDrive documents folder. I just have a thing called FlowJo plugins. And I just have all my jar files in there. RPath. So don't forget when you're putting your RPath, especially on Windows. So like here, this one went to my app data, local programs. So don't forget, you're going to go all the way out to this bin x64 folder. Okay, because that's where the executable that FlowJo is looking for lives. It's inside this x64 folder. When you run some of those R-based plugins for the first time, it's going to attempt to automatically install some of those packages. So the first time it runs, it might take a minute while it's installing a package. The next time you run it, it obviously won't need to install again, so it should run, calculate faster. One other tip, if you do have, it's like a very fresh install of R, is to just go ahead and try to install an R package, because you might get a prompt on your machine if it's the very first time installing a package, and it might say it's going to create, so it's going to create a folder to install R packages where you have read-write permissions. Because if maybe you're on a work computer, you might not have the permissions to install R packages in like the default location. So like if you go install.packages, and then you're going to install a package like PNG, and just hit enter. If I do it here, it's already installed, so we won't see the actual message that I'm describing. But if you were to get a message pop up saying, oh, I'm going to create a folder for you. Do you want me to do that? Just click yes. And it'll make sure that going forward, when you run a plugin and it's trying to auto install, that you shouldn't have any problems, and it's going to be able to install and put it wherever it needs. So I would say install R, try to install a package, let it create a folder wherever it needs to. Once that first one's good, you can go ahead and close R and then try to run a plugin. Okay. So what I have here, I've already gone through and run. So this is just I downsampled just a phenotyping data set to 50,000 events. Again, because I'm on this really small Windows machine. But it has something, I forget exactly, maybe like 38, 30, close to 40 different parameters, but it's just a high parameter immunophenotyping panel. And I ran a couple different embeddings. I ran tsne with like the default settings. Let's just take a look at tsne. I excluded some markers here because I didn't want to get into like really fine local structure of the data. I wanted a more less detailed embedding just for the purpose of this kind of webinar, to show some of these next things we'll be talking about. But like here's those iteration, perplexity, learning rate. These are, if you're on OPSNI option, um, this is going to basically try to tune those for you as it's running. Otherwise, you can go manual and try to adjust some of those things on your own. It is nice just to go with the OPSNI here. You have a couple different K nearest neighbors algorithm. So if you have a large number of parameters, millions of events, you might want to go with the approximate random projection or the annoy method, because that'll calculate a little bit faster. And then same thing for the gradient descent. You might want to go with the FITSNI if you have, again, really large number of events, high number of parameters. These can speed up t-SNE calculation a little bit. UMAP. UMAP. So you have some different distance metrics. Oh, I guess I'm realizing this wasn't even on my slides. But yeah, different distance metrics. So how it's, when it's building the nearest neighbor graph, like when it finds a neighbor, like the metric that it uses to consider something a neighbor or not, there's different, typically Euclidean distance is most often used, but there's some different methods here. Another cool feature with this newer version of UMAP is if you have a previous UMAP embedding and you load in like a new FCS file, you can go apply on map. I'm going to apply my new data set to this previous embedding. So it'll map the new data to this previous UMAP result that we see in the background. So you should get comparable, almost identical looking maps. Caveat there, right? You would need the exact same parameters exist in the two files if you wanted to do apply on map. But this is a good way, for example, if you had some really huge number of events, say you had a file with like 10 million cells and 30 different markers. Well, you might try performing a downsample, running UMAP the first time on a downsampled population. And then once you have that first embedding that runs quickly on your downsampled population, go back, select your 10 million events, and then say, I'm going to play on map from my first embedding. And that already has some information calculated about the nearest neighbors, the distances, things like that. And it can allow you to do that calculation even on a huge number of events. So that can be a way to get around an issue maybe if you don't have the amount of RAM available on your computer. Okay, so we got some embeddings, and then I went and ran FlowSum and Phenograph clustering. And what we see here, I've overlaid at the top just some of my manual gates. So like naive, what else? Some of these NK subsets somewhere down here. Different T cell subsets. So I've made those overlays here on both t-SNE and UMAP. Then I ran Phenograph. I got back 20 clusters, and then I went to FlowSum and I said, all right, now you give me 20 clusters. And now I've overlaid those two sets. Here on the left are FlowSum clusters on my UMAP and t-SNE. And these are Phenograph on UMAP and t-SNE. And there's this really cool tool I wanted to show you guys. It's called Sleepwalk. It's an R package. It's not available yet in Flojo. I said yet because I want to build this into a plugin for you guys because I think it's pretty cool. But it's a great way to kind of get an idea and explore your embedding. So, right, you get an embedding, but how do you really know? Some people might want to go and draw a gate on an embedding, but as you might know or suspect that how do you really know if an embedding faithfully really represents the high dimensional data in low dimensional space? Because I mean, it's trying to do a lot and embed all that information in just those 2 dimensions. And so with Sleepwalk, we can kind of look at that information in this way. So like on this side here, we have the UMAP overlaid with the clusters. And then on the right is the Sleepwalk tool. And basically what it's doing is when I hover my mouse pointer on a point, what it's showing is, here's a scale across the bottom. And where my pointer is, like that point, the color scale, when I hover, is the distances calculated from all these events in high dimensional space, but shown on 2 dimensions here. So in this way, we can kind of interrogate and move around our embeddings and get an idea of how well how well did it really embed my data? So like on the left, we have B cells. And I can see if I hover, this group looks pretty good. There's, for this area where I'm hovering, in high dimensional space, these events are also closely related to one another. You can see out on the edges where there's the pinker color, redder color, so those are further away. Strangely, if you go to this tip here, you see even in these B cells, for this particular point, there's this area where the CD4 8s, Ts, 4s, and 8s T cells light up. So what I did was I took these embeddings here that I ran on this data set, and I generated the same type of plots with that sleepwalk. Oops. So let me just scoot this over. And so these are the same UMAP. I have UMAP on the left and TSNE on the right. So if we hover over and kind of look at this area here, these are my naive CD4 positive cells. And we can see like in UMAP space, like when I'm hovering, we see that things are pretty tightly packed in this area when we're talking about the high dimensional distances of the events. So this gate, or at least like, so this embedding where it's separated these 2 kind of groups of events seems to agree well with the distances of the data in high dimensional space. When we get to the sort of center, things start to blur and spread across. But when we get up on this kind of other lobe, again, there's pretty tight grouping of events, at least in UMAP. But look over in TSNE space, you can see things aren't as kind of tightly packed and then spread around somewhat. And I found this other interesting area. If we look, so here in UMAP and TSNE, we have NK, mature NK. And with FlowSum clusters, it kind of split that into 2 different clusters. I wasn't able to gate that apart. with what I had done previously, but the clustering separated that area along with in t-SNE space as well. So if we go over and look on this t-SNE side here, so we're in the NK. What cluster is this? Like it might be 16 and 20. But anyway, so in t-SNE space, when I'm hovering over this area of these NK cells, I can see like in the t-SNE embedding, right, there's some more like definition here. And there's more local structure captured. And there's these tighter islands, at least in the embedding space. But when I hover over it, and I'm looking at now these distances in the coloring and the embedding, I can see that it's really like spread across this island, where on UMAP space, we lose some of that fine grained local structure, right? We do kind of see that there are maybe this lower half of the lobe agrees with what we see in high dimensional space. And once I come up here, we kind of only see events highlighted in this upper region. So that would tell me like, I agree with kind of these clusterings we get from flow sum, at least on this UMAP embedding. t-SNE is more complicated. Like there's all this additional local structure that probably, if we did some additional clustering, subclustering on these, we might pull apart some more detail that t-SNE can find, but the clustering didn't seem to capture. Anyway, really cool tool. I hope to build this into maybe a separate tool or maybe a feature added on to some of these embedding tools to really kind of get an idea of how well does the embedding represent my data, and then comparing that to your clusters. Again, it's called sleepwalk. There's some other methods for sleepwalk. One's called K nearest neighbors sleepwalk. You get the same type of visual, but it shows you K nearest neighbors distances instead of Euclidean distances here. However, the K and N sleepwalks pretty computationally intensive, and it tends to crash a lot. This one works better for me. So I think that's about it for my webinar. So if there's any other questions, feel free to type that into the chat. So someone's asking, I think here in Sleepwalk says, when I'm comparing and see differences in t-SNE versus UMAP, is there a sort of hierarchy of believability? Yeah, good question. I think it's not to say that, one's better than the other, or that I should believe one more than the other, just that depending on your data, one tool might do a better job at visually representing like what you want to show in your data. So it can help you interpret your manual gates, and then the embeddings, like how, like maybe, and again, there's other methods, right? There's tri-map, there's pack map. So some of those might really pull apart and show some local structure that maybe you want to dive deeper into with your particular data. So it can just help you understand if one embedding might do a better job of capturing that type of information that you really want to show to people. But yeah, comparing kind of the clustering with the different methods of dimensionality reduction. And then there's other cool things it can do too. Like instead of just showing different methods, like maybe you um, like run the embedding on multiple different samples. So you can, I think there's an example. So like you can show like here's 3 different samples. And you can see how those are compared to one another. This one doesn't have the same embedding, so it's kind of makes it a little bit harder to interpret. But with UMAP, like I was saying, there's that apply on map. So you could get all the samples onto the same embedding and then split them apart based on these samples. And maybe the samples are like different stim conditions, and you can kind of view the results that way. And if you do anything like with single cell RNA-seq or Seurat package, you can use this sleepwalk tool as well. Anyway, I thought that was pretty cool. So some resources here. There's our docs.flojo.com. It's a great place to go to look for any questions on Flojo, searchable documents. Flojo University has some short tutorial videos. And of course, there's where you can get the webinars. So learn webinars. Pre-recorded is all the pre-recorded webinars. So this one should be up there maybe next week. And yeah, feel free to reach out to flojo@bd.com. If you're looking for the slide deck sooner rather than later, I can try to send it over. So I have another question, but thank you guys for joining. Feel free to type in any questions. Someone's asking, what is best practice to concatenate files together to compare the pattern of immunophenotyping between controls and treatments? Yeah, good question. If you want to compare things like that, so you'll probably start out with your individual files in your workspace, and you'll want to leverage some keywords. So you'll create keywords that will identify those different controls and treatments, and then include those when you do the concatenation step. And then you might go through the traditional high parameter workflow where you're going to do dimensional line reduction and clustering, and then you'll take that into Cluster Explorer. And with Cluster Explorer, you can leverage those keywords as long as you've gated those apart in the concatenated file. You can load those keywords essentially into Cluster Explorer and then make some comparisons real easily that way. I'm trying to think. So there's probably some examples in our docs page. With Cluster Explorer. I'm not seeing it right offhand. The other one, let's check Flojo University. There might be like a workflow there that will go through it. So high parameter, no. So yeah, I don't see any short videos. So then where you're going to find it is if you go to learn webinars and then recorded webinars, and then or look for some of these like high parameter or like advanced Flojo webinars. And those will walk through like creating those keywords, doing the concatenation, dimensionality reduction, clustering, and then pulling all that apart in Cluster Explorer. Um, Let me see if I can find it. Otherwise, you could email flojo@bd.com and I can try to find a good webinar and send you the link. I don't see it offhand, but I can ask the group and they can point me towards a good webinar for you. But these are the resources basically, webinars, Flojo University. Well, thanks again, everybody. Like I said, feel free if any questions, reach out, flojo@bd.com. Right now, there's only a couple of us, so we'll get back to you as soon as we can. Thanks again for joining. Bye.